So the next topic that we're going to talk about before getting into meiosis, which is another type of cell division that happens in eukaryotic cells, is cancer. And the reason that we talk about cancer now in the course is because a lot of the genes that are involved in causing cancer are genes that are involved in some way in the cell cycle. Okay, so basically a lot of the genes that are mutated that end up causing cancer are genes that are involved in the cell cycle in some way. So as it says on the slide, about 1 in 5 of us will die of cancer, so that's one obvious reason to study cancer. And then another reason to study cancer is that it has led to some things about cell biology that we didn't know before. And specifically that's referring to the cell cycle. So we've learned about certain proteins or certain genes that are involved in controlling the cell cycle because we first found out that they were mutated in cancer, and then after we found out they were mutated in cancer, we discovered what they did, and we found out that they had a natural role in regulating the cell cycle. So it's led to some important discoveries in terms of finding out how the normal process of cell division occurs, and also some other processes. So there's two heritable properties that are important in terms of cancer cells. So they reproduce in defiance of normal restraints on cell growth and division. And so what that means is that what we'll see when we go through the properties of cancer cells is that there are signals in your body that tell a cell not to divide. And cancer cells are relatively insensitive to those signals, meaning when they receive the signal not to divide, they ignore it. Also, there are normal restraints where if you have DNA damage, that's going to halt the cell cycle. Cancer cells are also relatively insensitive to that response of DNA damage and halting the cell cycle. So rather than halting the cell cycle, they will just continue on through the cell cycle to produce more cells, even though they have DNA damage. And then a second heritable feature of cancer cells is that they invade and colonize territories normally reserved for other cells. And the process of moving to another site in the body, that's what metastasis is. So when you hear the word metastasis, that's the cancer moving from the primary site where it originated to a secondary site within the body. So basically what you have in cancer is you have uncontrolled cell division. Okay, so you have a cell that's going to divide to give rise to many cells that form a tumor. And then one of the things we'll see is that within a tumor, in addition to the cancer cells, you also have a bunch of normal cells that are recruited into the cancer. Some of those normal cells are helping the cancer to grow, and other cells can be there to combat the formation of the tumor. Okay, but these are two important heritable features of cancer cells. Just really quickly, in terms of the difference between a benign and a malignant tumor, the only difference that you need to know for this class is that in a benign tumor, the cancer cells have not invaded the surrounding tissue, whereas in a malignant tumor they have. So what they're showing you here is a normal breast duct, which is normally lined by epithelial cells. And you can see in this case all the tumor cells are confined within the duct. And then once it breaks outside of the duct and invades the surrounding tissue, then it's a malignant tumor. So as long as it's confined within its original site, it's benign. Once it starts to invade into the surrounding tissues, then it's malignant. So let's talk about first the causes of cancer. So we kind of defined two heritable features of cancer cells. So a majority of cancers are caused by mutations to the DNA of somatic cells. So we haven't defined somatic cells yet, but a somatic cell is any cell of the body that's not a germ cell or a gamete. Okay, so a somatic cell is any cell of the body that's not a germ cell or a gamete. So basically skin cells, neurons, muscle cells, heart cells, etc., all the cells that make up the different parts of your body are somatic cells. Soma means body. And so cancer is caused by mutations in the DNA of those cells. And then also there's things called epigenetic changes that are known to play an important part in the development of cancers. And in terms of, so genetic changes would be changes in the DNA sequence. So that's what a genetic change is. And genetic changes are heritable. So if you have a genetic change in a cell, when that cell divides and gives rise to two cells, each cell will have that genetic change in them. So that's heritable. An epigenetic change is a change that's heritable, but it's not due to a change in the sequence of DNA. Okay, so it can be inherited, but it's not due to a change in the DNA sequence. So those types of changes are also known to play a role in developing cancers. And then there are a few viruses that are also known to cause cancer. And on the next slide, it just gives you examples of that. So these are, you don't need to know any of them, but it just gives you examples of viruses associated with human cancers, and then specifically what a cancer they're associated with. Okay, so most cancers are going to derive from a single abnormal cell. And one thing that's important to remember for cancers is that, so you start off with a single abnormal cell, but that single abnormal cell used to be a normal cell. And you don't go from a normal cell into a cancer cell through one mutation. It requires multiple mutations in order to go from a normal cell into a cell that is going to develop into a tumor. Okay, so again, cancer requires multiple mutations, not just a single mutation. Okay, multiple mutations are required for cancer, not a single mutation. And what is going to happen when you have tumor progression is, and this is trying to highlight that, is you're going to have, this is going to be your normal cell that turns into an abnormal cancer cell. And what will happen is that cell will get one mutation that gives it a proliferative advantage over cells next to it, so it has a cell division advantage. And then one of the cells within that will get a second mutation that gives that cell an even more of an advantage in terms of cell division. And then that cell will proliferate more than the other cells, and then they acquire a third, and then a number of mutations. And basically each time that they acquire a mutation, that gives that cell an advantage. And then in the end, you're left with a cell that has multiple mutations that's capable of forming a tumor. So basically that's what I mean by this over on the left, that you're going to have several mutations, and after each mutation you have natural selection selecting for that cell that has a growth advantage compared to its neighbors. Okay, so we can talk about some of the properties that are associated that a normal cell has to acquire in order to be a cancer cell. And these properties are going to vary depending on the cancer that you're talking about, but these are a set of properties that cancer cells have that make them different from normal cells. So I'm going to go through them point by point. And so the first one is that they are more self-sufficient than normal cells for their growth and proliferation. So what that means is that the growth and division of normal cells is dependent on extracellular signals and on adjacent cells next to them. Cancer cells are more self-sufficient than that. They don't depend on extra signals in order to divide. They're also relatively insensitive to anti-proliferative extracellular signals, so I already mentioned this one to you. So there are signals in your body that tell cells not to divide at a certain time, and cancer cells are relatively insensitive, which means that they don't listen to those signals that tell them not to divide. So rather than not dividing, they would just continue to divide, even though that signal is present. Another important property of cancer cells is that they're less prone than normal cells to undergo apoptosis. So remember, apoptosis is a form of cell death. One thing that cancers are able to avoid is undergoing apoptosis. And so they're less likely to kill themselves than normal cells. They're defective in intracellular control mechanisms that normally stop cell division permanently in response to stress or DNA damage. So what that means is in a normal cell, if the cell is under stress or there's DNA damage in the cell, that we talked about, that's something that could halt the cell in a particular phase of the cell cycle. And cancer cells are defective in those mechanisms that will stop the cell cycle in response to things like stress and DNA damage. So they would continue to divide in the face of DNA damage, or in the face of stress, they're going to continue to divide. Also, cancer cells can induce the help from normal stromal cells in their local environment. So a stromal cell in a tumor is any cell that's not a cancer cell. So I told you that within a tumor you're going to have normal cells within the tumor. So within the tumor, any cell that is not a cancer cell is called a stromal cell. And some of those stromal cells are there and they actually help the tumor to grow, while other stromal cells are going to be doing the opposite. So they're going to be preventing or trying to prevent the tumor from growing. So stromal cells can be positive or negative in terms of their effect on tumor growth. Also, cancer cells are able to induce a process called angiogenesis. And angiogenesis is the growth of new blood vessels by sprouting from pre-existing ones. Okay, so angiogenesis is the growth of new blood vessels by the sprouting of pre-existing ones. So cancer cells, like any normal cell of your body, need a blood supply because they need oxygen and they need nutrients in order to divide, just like any normal cell does. And if a tumor's going to grow past a certain size, meaning if it doesn't induce an angiogenesis, it can only grow to a certain size, and then it can expand farther than that if it induces angiogenesis. And what happens is blood vessels grow into the tumor, and then they feed the tumor cells, and the tumor cells are able to proliferate more, and that causes the tumor to grow in size. Also, cancer cells are able to escape their home tissues, so that's when they become invasive, so they invade the surrounding tissue. And they're also able to survive and divide in foreign sites, and in that case you would say that the cancer has metastasized. So metastasis is when the cancer moves from the primary site where it originated to a secondary site in the body. Also, cancer cells are genetically unstable. So what that means, that's referring to DNA. And to say that something's genetically unstable means that it's going to acquire changes to its DNA at a higher rate. Okay, so what that means is that cancer cells will obtain DNA mutations at a higher rate compared to normal cells because they're genetically unstable. And then this one's not going to make much sense right now, but it will later on when we study DNA replication. The ends of linear chromosomes, and remember that chromosomes in eukaryotic cells are linear, are called telomeres. And what happens is every time a cell divides and you replicate a linear chromosome, the chromosome loses a little bit of DNA at the ends. And so what that means is every time you replicate that linear chromosome, the chromosome is going to become shorter and shorter. And that can only happen to a certain extent, and then the cell's going to stop dividing. Okay? And what happens in cancer cells is they either produce this enzyme that's not normally active in most of your somatic cells. It's called telomerase. And what telomerase does is it lengthens the telomeres. So even though they might be shortening, this will lengthen them to keep them from becoming too short. Or they have some other way of stabilizing the ends of their chromosomes so that the telomeres don't shorten. And that's also an important characteristic of cancer cells. So again, these are all properties of cancer cells that are going to differentiate them from the normal cells that surround them in the body. Okay, so I told you that previously that cancer is caused by mutations to the DNA, and then it's important to know what are those mutations affecting to cause cancer. And so there's two types of genes in terms of classes that are involved in causing or being critical to cancer. One type is called proto-oncogenes. So proto-oncogene would be the normal copy of the gene, and then when it becomes mutated and it's a cancer-causing gene, then it's an oncogene. So the proto-oncogene is the normal gene, and the oncogene is the mutated cancer-causing form of the proto-oncogene. And then you also have a class called tumor suppressor genes, and we'll talk about why they're called that in just a second. So the definitions for each of these are on the next slide, and so you do need to know them, and I'll explain each of them. So a proto-oncogene, again, is the normal gene, and proto-oncogenes, as it says on the slide there, are usually concerned with regulation of cell proliferation, so regulating whether a cell is going to divide. And a proto-oncogene can be mutated and form an oncogene. So as an example, let's say that the normal function of the gene is to code for a protein that when it's active pushes the cell into the cell cycle. So it pushes the cell to divide when it's active. And so one thing a mutation might do to cause it to become an oncogene is that the mutation causes the protein to always be active. And so what that means is that now it's constantly telling the cell divide, 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 rather than waiting to be told when it's supposed to be active and when it's supposed to divide. It's constantly on. So then an oncogene is an altered gene whose product, and I'll explain this in a second, can act in a dominant fashion to help make a cell cancerous. Okay, so I told you previously that the cells of your body have 2 copies of every chromosome, which means they have 2 copies of every gene. So let's say that you have 1 copy here. So the rectangle represents the proto-oncogene, and then the other chromosome carries the oncogene. Okay, so the phenotypic effect of the oncogene is that it causes cancer, and we'll just say the phenotypic effect of the proto-oncogene is that it doesn't cause cancer. And what it means that it acts in a dominant fashion, it means that if you have these 2 copies present in the same cell, the phenotypic outcome of this gene dominates over this one. So if you have one gene that doesn't cause cancer and one form of the gene that causes cancer, the form of the gene that causes cancer is going to dominate and cause the tumor formation, or make it more likely for tumor formation, dominate over the one that doesn't. Okay, and what we're going to see is tumor suppressor genes act in a different fashion. Okay, so tumor suppressor genes are genes that appear to help prevent formation of a cancer, and these genes can have several functions. So some functions of tumor suppressor genes are code for proteins that inhibit the cell cycle. Okay, so if a protein was involved in inhibiting the cell cycle, that obviously would help in preventing a tumor from forming. Also, some tumor suppressor genes are involved in apoptosis, so causing a cell to commit cell suicide. And then other tumor suppressor genes are involved in repairing DNA damage. And in terms of this one, typically it's a loss of function that's going to favor the development of a cancer in these types of genes. So I previously told you for a proto-oncogene, you can go from it being normal into a hyperactive form, so it's always on, so that would be enhancing its function. And tumor suppressor genes tend to lose their function, which makes sense because if you have something that's supposed to prevent cancer formation, losing its function means that you would lead to cancer. Okay? And typically for tumor suppressor genes, both copies have to lose their function. Because as long as you have one functional copy of the gene, you're fine, but then when you lose both, then that's going to lead to cancer. So typically for tumor suppressor genes, you have to lose the function of both copies of the gene in order to have cancer caused. In terms of, so you have proto-oncogenes and also tumor suppressor genes that are involved in regulating the cell cycle. And with respect to that, you can think of oncogenes in terms of causing cancer, you need 2 things to happen. You need the signals that tell a cell to divide to be on, and you need the signals that tell a cell to stop dividing to be off. Okay, so you can think of oncogenes as the gas pedal in a car, and you can think of the tumor suppressor genes as the brake. Okay, so if you want the car to go forward, you have to have the brake off and you have to have the gas pedal pushed down. So you push down on the gas pedal through oncogenes, and you release the brake by mutating tumor suppressor genes. Okay. These, you don't have to remember anything from this slide. These are just two tables taken from your textbook giving you examples of oncogenes and examples of tumor suppressor genes. The oncogenes list doesn't really tell you much. It just tells you what the function of the protein is, or sorry, what type of protein, not its function, what type of protein it is. But for the tumor suppressor genes, it tells you what they're involved in. So when you read through the list, you can see that some of the tumor suppressor genes are involved in apoptotic pathways. Other tumor suppressor genes are involved in repairing DNA damage or detecting DNA damage, whereas other tumor suppressor genes are involved in negatively regulating the cell cycle. So a loss of function in those types of genes can all lead to tumor formation. Okay. So again, just as an example of a cancer-critical oncogene, so this is a gene that's mutated often in cancers, is RAS. So RAS can be mutated to be an oncogene. And if you remember back to cell signaling, RAS was part of the pathway where you had EGFR, the epidermal growth factor receptor. That ends up activating RAS. When RAS becomes active, that eventually leads to activating transcription factors that alter the expression of genes involved in the cell cycle. So basically activating RAS leads to activating the cell cycle. And so mutations in RAS that make it hyperactive or active all the time can lead to causing cancer in a cell. And RAS is frequently mutated in cancer. Another cancer gene before we get into that, another type of example of a specific tumor suppressor gene is this gene right here, RB. That's called retinoblastoma. So retinoblast, blastoma. And what they're showing you here is, again, this is a signaling pathway, and it's showing you that retinoblastoma inhibits this transcription factor called E2F, and it's showing you that E2F causes the transcription of genes that control entry into the cell cycle. Okay? So if you inhibit this, that's going to prevent entering the cell cycle. So when RB is active, it inhibits progression into the cell cycle. So retinoblastoma would be a negative regulator of the cell cycle. So it's loss of function mutations that are going to turn RB into a gene that causes cancer. So that's an example of a tumor suppressor gene. And the natural function of this tumor suppressor gene is to inhibit the cell cycle. And it does it specifically by inhibiting this transcription factor that regulates the expression of genes involved in the cell cycle. And then just to build on top of that, you can see that retinoblastoma is actually controlled by a cyclin-dependent kinase and a cyclin D CDK4 complex. This complex, when it's active, inhibits retinoblastoma, and then if retinoblastoma is inhibited, that means E2F is active. Okay. And then cyclin D or CDK4 are also known to be mutated in cancers, and they're oncogenes. And then you can see above that, you have this protein called P16 that negatively regulates the cyclin-dependent kinase. And P16 is an example of a tumor suppressor gene that you can find mutated in cancers. And so if this is mutated, then that means that this complex is going to be active, which means that it inhibits this. A loss of function of this means that that's going to be active and pushing the cell to go through the cell cycle. Okay. And so that's an example of a pathway that in the cell that actually involves multiple genes that are mutated, are found to be mutated in cancer. And then another frequently mutated cancer gene is a gene that's involved in regulating apoptosis. And in this case, the name of the gene is P53. And there's different things that can activate P53. And then one of the things that P53 can lead to is apoptosis. So if you mutate P53, that would be a way of specifically making a cell insensitive to apoptosis, or less likely. That cell is going to be less likely to undergo apoptosis if it has a mutated P53 gene. Okay, so that ends the cancer part, and so we'll go through some of the lecture questions for this topic. Okay, so in terms of number 3, what I'm asking is, what causes most cancers? Mutations to the cell's own DNA. Okay, meaning there are some cancers that are caused by viruses or that are helped along by viruses, but most cancers are going to be involved by mutations to the DNA of the cell. Number 4 is important. Does a normal cell get converted into a cancer cell by a single mutation? Right, no. And then 9, mutations in genes that regulate what process can allow cancer cells to survive when they should not? Right, apoptosis. And then do you guys see other questions from this lecture questions list that you guys want to go through? Yeah.
I just have not really two questions. With radiation treatment, does that target at this time?
DNA damage, and then that'll cause the cell to cause cell death. Yeah. Yeah, radiation. And also radiation is non-specific. So the reason you lose your hair with radiation treatment is because it'll also kill the cells that give rise to hair are dividing, and so those cells are also going to undergo apoptosis, and you basically kill those cells off, and that's why you lose your hair. And so basically radiation is going to affect any dividing cell within your body, which is why there's all these side effects to radiation. That's true for other chemotherapy drugs. A lot of the first chemotherapy drugs that we have target proliferating cells, which means they're going to target any dividing cell, and they're not specific for the cancer cell. And so basically the drive now is to come up with targeted therapies that will target a cancer cell and not normal cells. And basically you're trying to target the different pathways that are involved in causing a normal cell to become a cancer cell. So that's another reason that it's important to study cancer, is then, okay, maybe I can inhibit this protein and that will inhibit the cancer cell but leave my normal cells okay. Do you have another question? No. Okay. Other questions? Okay, so because we're not going to get significantly farther into the next topic, what I want you guys to do is to bring your notes to class on Wednesday, so bring them to lab, and we're going to do a half hour of lecture in lab on Wednesday. So bring sexual reproduction and meiosis, bring that PowerPoint with you to lab on Wednesday, and we're going to do some lecture, half an hour of lecture in lab. So now what we're going to do is we're going to stop now and we'll go over to the lab, and you guys are going to set up your second seed research project.